Hello. The nuclear genes are numerous, and each of these genes have numerous exons, which would also mean they have numerous introns, and the introns can be very long. And yet, the splicing of the exons should happen in a very coordinated and precise way. Otherwise, there would have been a lot of waste of energy, matter, and also the time. For these, we have dedicated Spliceosomes, the machinery that actually performs the splicing, and it has a ways of somehow increasing the fidelity and also enhancing the way the reactions happen. That's what we'll have a look at now. So there are several factors that are required for the transesterification reactions to happen, and that means we would also have and there are several proteins as well as several RNAs that are involved, and it requires energy for the splicing reaction to take place. The RNA components have a special function to recognize the intron-exon borders and also the catalysis. The RNA components, because by using the base pairing, they are able to recognize the 5' splice site as well as 3' splice site, and also some of the interactions with the branch site as well. That is where the catalysis, we will have a short look in a short while. They are called as small nuclear RNAs, which are usually about 100 to 300 bases long. The proteins, when they are in complex with proteins, they perform these functions. It's almost like the enzymatic reactions are performed by the RNA components. It's the same case with when you see about the ribosome during translation. The RNA components have serious functions that are essential functions to make these things happen. So how does fidelity happen? One of the points is that for fidelity, you can have the bases, and although we have designated that these bases are they recognize the sequences here, but it's not always the same thing. What I mean is, some of these splice sites could differ in their sequences. So the splice site may not match absolutely like what is shown here. So there are several mechanisms to increase the probability of accuracy or fidelity. That is, say for example, U1 is one of the splicing machineries, SNRNP, and which recognizes the 5' splice site, that is GUA/GU, through the RNA component that it has. So it base pairs exactly, and it is U1 is replaced later in the stage. At a later stage, it is replaced by U6, which also has a RNA component which can bind, if not as perfectly as U1 does. So having two different components to recognize the same point would increase the probability of accuracy, because maybe U1 has made a mistake, but U6 may have covered it up for it, something like that. And there is also another function for, say for example, U2. We have this branch site adenine, which is important, whose 2' OH is going to make the first transesterification reaction. So if you look at closely the sequence that binds to the branch site, we will notice that the sequence, this is 5' to 3', so I'll just read from here. That would be the 5', GUA, GUA, and it perfectly base pairs with the region at the branch site, except for this adenine. So the branch site adenine is UAC, UA, and there is an extra A and then C. So basically, when these two base pair, you'll have one adenine that is not base pairing, which is probably flipped out, and then its 2' OH is activated to perform to carry out the first transesterification reaction with the 5' splice site. which will allow the formation of the 2' to 5' linkage. And there is also another RNA, another component, that's the U6. U6 has to, say for example, U1 has recognized the 5' splice site, and U2 has recognized the branch site, and the transesterification should happen, which means that the 5' splice site should come in close contact or proximity of the branch site, and that is mediated by U6. If we remember, U6 is binding to the 5' splice site, and it's also interacting with the U2, which means these two the points are brought together by U6. There is also similarly a protein called as branch point binding protein, which recognizes the branch point first. It is just protein, and then it is replaced by U2 to bind here, and then adenine gets activated. So as we can see that the RNA components play an important role in the splicing itself. Here they are recognizing U1 and U6 are recognizing the 5' splice site. Branch site is recognized by U2 and BBP, and U6 is bringing the branch site and 5' splice site to close proximity. And we also have U2AF 65 and 35 others that recognize the 3' splice site. Let's have a quick look at how the sequence of steps that happen. So the first, we have the U1 binding on the 5' splice site, and BBP, branch point binding protein or branch site binding protein at the branch site. And here we have the U2AF 65 and RS35 and 35 that bind the 3' splice site. So BBP is replaced by U2, and U2 through its base pairing will allow the flipping of one adenine that doesn't base pair, and the 2' OH of that adenine will get activated at a later stage. Now there is a bigger multi-protein complex with RNAs that's called as U4, U5, U6 snRNP that binds to U1 as well as U2, both at a time. So which would mean that these two are brought together into close proximity, and then the adenine would get activated and will make a transesterification reaction with the 5' branch site or splice site, and U2 so U4 is released out, and the 3' OH of the exon would make the second transesterification reaction, thereby resulting in the splicing of the exons. Here we have the two exons spliced together, and the lariat along with the U2, U4, U5 and U6 is released out, which will then be degraded. And the splicing machinery proteins will go back to splice another set of introns or allow the removal of the introns and splicing of the exons. Thank you very much. We'll see you later in a different talk.